Well, as you enjoy your lunch, I'm going to walk you through, from a high level, what developability is at Catalent, and then take you through a case study where we evaluated developability and share some data. In Kansas City, Catalent's a global network of various sites, but in Kansas City, we do both small molecule manufacturing and we're also engaged in biologics. We're a full-service analytical lab. We also, within the network, manufacture drug substance and drug product. Although that doesn't happen at Kansas City, we're strictly an analytical lab. So great on-time delivery, you get a dedicated tech lead and project manager. We have over 30 years of experience there with biologics. We have a significant number of our analysts have advanced degrees, and we support a lot of programs. Our last FDA audit, we had 0 483s. We usually have about 30 audits every single year, whether they're client or regulatory. We have about 200 scientists, and square feet and cubic feet don't mean a lot to me. So I actually looked up how much 20,000 square feet of lab space was, and it turns out it's about 10 tennis courts. And then we have 31,000 cubic feet of stability storage, and that ended up being about 20 shipping containers. So quite a bit of space analytically and storage-wise. We support all kinds of modalities, mAbs, pegylated peptides, ADCs, conjugates. We also do cell and gene therapy, nucleic acids, fusion proteins, and we support those from discovery all the way through commercial. Like I said, we're a full-service analytical lab, so I'm not going to go through the long list. I'm a scientist, and these are very salesy, so I apologize. Okay, so getting into developability. What is developability? Well, like I said at the beginning, it's a way to de-risk your asset development. You want to get an early read on what developability is. And today, in the green boxes is what I'm going to cover. So, some of the things that we do up front, we're trying to predict whether, the molecule will be stable. And the way we do that is via solubility testing, colloidal and conformational stability, and we also use in silico modeling. So we're trying to predict whether or not, if you have multiple candidates, which of them would be, ones you would want to be your lead and take forward into development. We also, I'm going to talk today a little bit about formulation. We did a screening for formulation of an antibody, and then along with that, we also did some forced degradation. So things I'm not going to talk about today, but also are a part of developability is another not real word, manufacturability, immunogenicity risk. You can leverage in silico modeling, and earlier this year, the FDA gave a roadmap for reducing animal testing in preclinical safety studies. That guidance, I think, will start paving the way for more of, machine learning, AI, those types of tools as a surrogate for animal testing, and also, to help speed up the development process. So if you skip developability, what are the risks? Well, reformulation, shorter shelf life, you may have to do additional testing, and all of those things will spell, regulatory delays, increased cost, and, ultimately, there's a risk of project termination. So moving on to the case study, we chose a commercial mAb, cetuximab, as our model protein. We did that because of its significance as an IgG1 mAb, so easy to handle for the most part. And we understand, since it's a commercial product, it should be well behaved and fairly stable. But we wanted to see, if we evaluated it as part of a developability study, what we could learn, additionally from from our data. So starting with predictive stability, this part of the process is really about, it's not about finding the best conditions for your mAb or your protein or your modality. It's about eliminating poor choices. So you're looking for red flags. So the in silico piece, here are some of the things that we can do with the software. We do use a commercially available software. It has a library. We can evaluate known immunogenic moieties on your protein. We could do surface patch analysis. We could even do, if we know what the dock looks like, protein-protein docking. Essentially, the in silico will help guide us, as a paper exercise before we begin to, before our partners begin to invest money in their programs. So here's the design of our predictive stability. Now, you don't have to do this. For this study, since we only had one particular candidate, we decided to also combine the predictive stability along with some pre-formulation screening. So since we aren't looking to select a candidate here, combining it with the pre-formulation screening gave us additional insight on protein stability. And it helps inform some of our later experimentation. So what we did is we chose 4 buffer systems, 3 pHs each. And then across the rows, we chose a control so we could isolate just the buffer system. And then we also added arginine, sodium chloride, and sucrose to the formulations. Everything was formulated at 5 mg per mil. And then the tests that we performed, we evaluated melt temperature, we evaluated KD, the diffusion interaction parameter by DLS, and we also evaluated solubility. Speaking of solubility, the way we perform solubility, and I think it's an industry standard now, is you titrate PEG into your sample. PEG acts as a crowding agent, and it forces the protein interaction. And as you titrate up, you evaluate either precipitation, either loss of protein through precipitation by A350. I've seen other labs use A280. They pellet any precipitate and then measure A280. Here we used A350. And as you see, this is cetuximab in its native formulation. So looking closer at the data, we chose to use a QT or RT-PCR because all you really need is to be able to titrate in a hydrophobic dye and then have fluorescence detection. So for our DSF experimentation, here's some of the data. We didn't see, like I say, any real red flags across these different conditions. If you look at the sodium phosphate pH 7, no excipient, and also the one with sodium chloride, the melt temperatures are fairly high in comparison to some of the other wells. And that is closer to the originator's formulation. So it appears that, I guess they knew what they were doing when they formulated their commercial products. It looks like, at least for melt temperatures. For DLS, KD is the diffusion interaction parameter, and that what we're looking for is positive KDs. We want to see protein repulsion. When you have negative KDs, it indicates that your proteins are attracted to each other and will lead to aggregation. And then just for fun, we also did a thermal ramp, and then also evaluated KDs as we increase temperature. And that data corresponded pretty well with our melt temperature data on the previous slide. So what was the outcome? We didn't see any, like I said, we didn't see any red flags with the buffer systems. They were all okay. We saw some potential benefit from sodium chloride at low PEG concentrations. And as expected, we were also noticing that at a lower pH, the colloidal stability seemed to be improved. We saw a slight benefit from sucrose that was shown in our KDs. Didn't matter the buffer, didn't matter what pH. when it had sucrose present, it had a positive KD, so that was interesting. So we're seeing some benefit from adding the sucrose, or some, and as I'll show later, some of the other bulking agents seem to help with colloidal stability as well. So talking a little bit about forced degradation. In our, you want to try to evaluate your proteins as early on. And why does forced degradation matter? Well, you want to make sure that your protein is chemically, physically stable, and that you are assessing it for, future, again, red flags. Do you think there will be challenges as, later on? What can you expect? And it helps you refine your strategy for development, your regulatory strategy. So in the case study, we actually put it through its paces. We did hydrolysis, both acidic and basic, oxidation. We did a light study. We did thermal stress, freeze thaw, and then we did 2 types of stirring, mechanical stress. One was stirring and the other one was shaking. We have noticed that there's a difference between those types of mechanical stresses, so we wanted to evaluate both. And just from a high-level overview of the data, unsurprisingly, we found that thermal stress led to aggregation and acidic variant generation, and that the thermal and stirring stresses increased the average radii and DLS. We saw some clipping and also some covalent aggregates in our reduced CE-SDS. So kind of looping back now, so we started out with the predictive stability. We did some pre-formulation screening. Now we wanted to take it forward and refine the formulation for cetuximab. So we used the predictive stability data that we gathered, and it informed this. We ended up choosing, for this study, 48 different conditions, and we chose histidine at 2 different pHs, and we also chose phosphate. We chose histidine because it's commonly used. It's standard, almost like a platform for many companies to formulate their proteins in histidine. And the phosphate we chose because that's the originator's buffer, formulation buffer. So we alternated between arginine and sodium chloride to help with ionic strength of the solution. And we also, this was a full factorial DOE. So we also did alternated between the sucrose, sorry, it's really hard to see up there. It's hard for me to see down here too, I apologize. But we used sucrose and sorbitol. And then across the rows, we added in polysorbate. One row as a control with no polysorbate, and then we increased the concentration all the way up to 0.1%. Then we stressed the material, because we want to get definition now. Again, we didn't see any red flags. Now we're expecting that without stress, we may not be able to get differentiation between the conditions. So now we want to force degrade it so that we can get differentiation. So we did shear stress, 300 RPM for 3 hours. We did 5x freeze thaw, and then 4 weeks we did 5, 25, and 40 degree conditions. And then after performing the stress, we performed A280. This is all plate-based, by the way. It's really the only way to go when you're doing DOA experimentation, because it helps. If the method can be plate-based, it's super quick. A lot of companies are looking to have this developability assessment done, somewhere between as quickly as 4 weeks, but if you do like a full screen like this, up to 12 weeks. So within 3 months, we should have a pretty good idea of a final formulation. So after the data analysis, we did a holistic view of using heat maps. The legend shows that if it's a green check mark, that's great. If it's a red X, that's very bad. What we noticed was that if you look at the arginine, it just didn't seem to perform as well as sodium chloride across the board, especially with phosphate. I mean, essentially all the conditions have a red X except for one, which is also red. We also noticed that histidine seemed to be good for clipping and subunit purity. When we did our CESDS, we did both reduced and non-reduced, and we saw that it just performed better, the histidine buffers, than the phosphate buffers. We also noticed that sorbitol outperformed sucrose, and it performed well across, almost regardless of the pH and the buffer system that we chose. We got a little bit of mixed signals on polysorbate. We do see the originator doesn't have any surfactant in their formulation. Maybe the protein doesn't necessarily need it, but as kind of as a precaution, I think typically, if you have a platform formulation, you would want some type of surfactant just as a precautionary measure. So with all this data, I thought, when you look kind of at a cluster over here, we have 2 green check marks when you add in at least 0.05% polysorbate in the histidine buffer system with sodium chloride and sorbitol. And you see something very similar over here, same conditions, or same excipients, lower pH. So looking at holistically at the data, I think, a histidine buffer system, sodium chloride with sorbitol, and at least 0.05% polysorbate 80 would help stabilize this protein potentially better than maybe the originator formulation, at least in our testing. Because you can see down here, this is the originator formulation, didn't seem to perform well. We had trouble reconciling this data, though, with this condition, which is kind of similar in, as far as a formulation. But it could be that the bulking agent adding in sugar helped stabilize it above and beyond what the originator formulation had done. So maybe putting a bow on all this, I think early developability and formulation assessments are critical to help accelerate the development of your proteins or other moieties. I think it's important to avoid, again, making mistakes. It doesn't take the challenge out of developing your product. It just makes it so that you're aware of the risks and you can come up with a mitigation plan for those as quickly as possible. We see more and more that we're getting requests, and there's, I think, a future for sure with in silico modeling. The more that we model, the more we learn, theoretically about proteins and their stability, and can help predict whether or not it's something that could be easily developed. And I think, ultimately, this will help streamline the development of your products. Well, thank you for your attention. Hopefully your lunch was good. I'll take any questions now.
So I'm just curious if you can go back to that check mark, red dot, green dot, yellow dot, like a stoplight kind of situation. I'm curious what parameters you put into it. So the parameters that rolled into here were based on the 2 prior studies, which would be the freeze thaw and your initial developability study?
This data is just strictly has to do with the formulation definition study that we did. So this is from stress. So we did the initial screening, we learned something from it, then we move forward into this study. So the data here is isolated just to that. So we've moved past some of those early formulations that we had evaluated.
I see it. And this data is based upon what analytical technique, DLS or what?
So it's all the techniques that on the previous slide. So it's 280, 350, SEC, MFI, CESDS, reduced and non-reduced.
Got it, okay.
Yeah, and it does become a challenge, right? Because you can't weight some of the data, all at the same level. Clearly, like aggregation is something that you'd like to avoid, and so we did weight that one slightly higher. So we had kind of an approach to looking at evaluating the data, and it was a risk-based approach on the critical quality attributes that we were interested in.
But should A280 change, or it will crash out a solution? Is that what for example, you're measuring A280?
So 280 was simply to inform some of the other methods if the concentration had changed. And we didn't notice any gross changes in concentration across the conditions. And then 350 is essentially light scatter, right? We're looking for if there's any turbidity, subvisible particles,
Aggregation.
Yeah, aggregation in general.
Okay, thank you.
Mm-hmm.
Just really quick question. I know a lot of the studies you have done for protein formulation. Does some of these technology also apply to, say, viral vector formulation development as well? I'm not a formulation scientist, so it might be a dumb question.
Yeah, and it is a good question. And I think that, yeah, for viral vectors, I don't know that formulation, I think seems to be like a few formulations that just work well. I don't know how much work actually goes into developing a formulation for those types of molecules. And to be frank, we haven't handled a formulation project on our site for viral vectors yet, but we do get a lot of requests analytically for them. So I guess maybe it's just my assumption that maybe they're not that difficult to formulate.
Hi, nice presentation. So I have actually a couple of questions. So the case study you presented is a mAb. I assume in your portfolio you're also dealing with a lot of bispecific or other more complex design. Do you normally take a similar approach for those complex molecules?
Yes and no. It's kind of an iterative process. So mAbs are probably the types of molecules we handle the most, followed by ADCs, and then fusion proteins are probably after that with a couple of other types of molecules. But this process is kind of our standard process to take essentially all the molecules through. But based on what we learned from maybe the earlier experimentation, we may tweak the future studies. And again, it depends on what our clients are asking us to do. If they just simply have, a few candidates that they want us to compare to each other, maybe they don't even want us to do formulation work. It might just be that predictive stability and developability, like in silico work. Then they choose their candidate and, they move forward.
Okay, great. And another question, personally a big fan of predictive stability method, but at the end of the day, to nominate a formulation, I guess we need some real stability data. So how much stability data do you use to confirm a formulation, like 4 weeks, 8 weeks?
It depends. I would say at least 3 months is probably what you would want to do for, using your accelerated conditions on stability. So this is plate-based. Your prototype stability study then would follow after this. So you pick a lead, maybe even a backup formulation, right, and then you fill it into a vial or whatever. Maybe you have a good idea what your TPP looks like, and then you fill your final formulation into your device, and then you throw that on stability. But I think at least 3 months. Typically, we'll take those studies out to about a year, maybe sometimes 18 months, just to have confirmation, at recommended storage. Usually, a formulation lock can happen about after 3 months.
Okay, got it. Thank you.
Mm-hmm.
Thanks. So at what point in the developability screening do you move from DSF, or would you move from DSF to DSC?
That would probably come towards closer to the end when you're actually wanting to characterize. So it could be used either as part of your force degradation study, or you could wait until the end when you're doing the confirmation of your formulation. So you select a formulation and a backup. then you can take those formulations and do a much more comprehensive screening, analytical screening of your protein in those formulations.
Okay. And then my second question is regarding formulation. Do you guys look at the feasibility of lyo products? So, for example, having salt or sodium chloride in a product may make it or ruin cake appearance. So, do you consider that in your process of narrowing down formulations?
Yeah, and again, maybe this iterative approach, right? I mean, if a client knows I'm going to want to go, I'm going lyo because we know that there's, we have these liabilities on the protein that, we won't be able to go liquid formulation. It's going to have to go lyo. Then, if we know that from the beginning, then we could tailor, the screening to make it a lyo-friendly formulation.
Hi. I'm wondering, for the stresses that you looked at, did you look at light exposure and as well as other like transportation stress?
We did look, as part of the forced deg, we didn't evaluate light stress as part of the formulation development, but for the forced degradation study, we did light stress. We covered the entire range, at least for this protein. But again, it depends on the client's request. If a client wants to do more, we can do more. If they want to be a little more limited in their stress, we can do that as well.
Another one here.
Hi, nice talk. Could you speak a little bit more about the in silico analysis, particularly on how do you combine that patch analysis or whatever analysis you guys do with the high throughput screening data you have?
So is your question about our method for evaluating the data?
Right, for in silico analysis, or do you develop a predictive model, or how does these two components come together?
Yeah, we have a mass spec team that handles a lot of that. So I won't have a great question or an answer for your question, but we have commercially available software. I think it's actually quite expensive. So it does a lot of the work. I know that there's other options out there for modeling proteins and evaluating the sequence for liabilities. But we actually we subscribe to some professional software that has these libraries built in, and it essentially, you just drop in the sequence, it models it for you, and it will help identify some of these things. So if we may be talking a little bit more about what we observe during in silico modeling, that could be a flag for us. If we see large charge patches or hydrophobic patches, those could be things that we would maybe recommend, especially for evaluating multiple candidates. That would probably be on a lower tier as far as something that could be developed.
Any other questions? Okay. Well, Catalent are booth 718, is that right? Yep. So if you have any other questions, you can visit them there. But in the meantime, let's give John a big round of applause.
And as long as I'm up here, if I could, on the 24th, if you're interested in this data, we're going to have a webinar on September 24th. And there's probably 2 or 3 times the amount of slides that I had today. And I'll go a little bit deeper into the data, but technique by technique. So if you're interested in that. Also, poster presentation on Thursday morning. And again, I'll have a little bit more data than the summaries that I have up here. Thank you.